Hello and welcome to Understanding Biology.
Yay!
Today we will talk about what controls the cell cycle and its function. First, the steps to the cell cycle are as follows: G1, synthesis, G2, and mitosis. G1 is when the cell continues to grow at a normal rate and continues its everyday processes. Synthesis is when the cell takes time to replicate its DNA in order to be ready to split. Then, for G2, the cell continues to grow into its final stages before separation. The process of G1, synthesis, and G2 are known as the interphase, which you should have already learned through the other steps of mitosis. And finally, the mitotic stage, commonly referred as the starting point for mitosis and the overall duplication of the mother cell. In between certain parts of these steps, there are what we call checkpoints. These checkpoints make sure everything is functioning properly inside the cell. There is a checkpoint before synthesis and another one before mitotic stages, as shown here. If there is a problem with the cell when it reaches a checkpoint, then the cell would not continue with the cycle, it would stop. If it stops completely, this is called G0. But doesn't that have to do with physics or something? No, of course not. That's 0G. This is G0. Let's try to pay attention. Certain factors can help speed up or cause the growth of the cell. These are called growth factors. Wow, now that's easy to remember. It can be. An example of a growth factor is known as a platelet-derived growth factor. This growth factor is found in your blood and allows the cell to start its process and continue after the checkpoint. But this is not the only factor that affects the cell process. There's more? There is indeed. External factors like nutrients outside the cell and the cell's environment, along with internal cyclin and cyclin-dependent kinase. Teacher, what's that cycling stuff you said? Cyclin, not cycling. Cyclin is a protein inside the cell that builds up over time. As it builds, it starts to connect with cyclin-dependent kinase that is also found in the cell. The CDK are enzymes that are found around the cell and in that area. After a certain amount of cyclin attaches itself to its designated CDK, the cell then knows that it is time to start the rest of the cycle.
So what happens if there's no control over the cycle or whatever?
That's a good question. When cells divide, there is a strange phenomena called density-dependent inhibition.
That's a big word.
This occurs when cells continue to grow until the space they occupy is full. Then they halt their growth. However, if the cell doesn't have control over its growth, then that could lead to rapid cell reproduction, the same reproduction cancer cells go through. By not halting the growth process, cancer cells build into tumors that will continue to grow over time.
Yeah, that sounds really bad.
It really is. But through technology, we have figured out a way to control cancer before it starts to spread too fast. That's it for today. I hope you learned a lot. No homework, just go and study. See you tomorrow in class. You're all dismissed.